Hi, this is Group 8, the impact of highways on arboreal biodiversity. Our introduction was that human infrastructure causes habitat fragmentation, and habitat fragmentation tends to lead to overall lower biodiversity. Our question was, how do Michigan interstates affect biodiversity of tree species in an ecosystem as measured by the Simpson's Diversity Index compared to non-highway impacted ecosystems?
So for our methods, we went to 8 different locations and we measured out one big 100-foot transect, and then we took 10 smaller 10-foot transects within that, and we took pictures of trees along those transects. For our statistical methods, we use a Simpson's Biodiversity Index to measure the biodiversity of our different sites, and we used a t-test to determine if there was a difference between the sites.
For the results, we have two graphs. The first one is the biodiversity per site. So for example, HW1 is Highway 1 and RA1 is Rural Area 1. And the other graph we have is highway and rural biodiversity, which is the averages of the highway biodiversity and the rural biodiversity.
Okay, so for the discussion, we can conclude that the presence of highways do not greatly impact overall biodiversity. These findings are contrary to established research that we've looked at prior to this, but we know that this can't be applied everywhere. And then possible errors in our assessment could have included incorrect transect measurement or incorrect tree identification.
For the take-home message, even though there are structures that have negative impact on ecosystems that we saw in previous readings, we could not find a significant difference in biodiversity between forested areas and highway impacted forested areas in Michigan.
Although it is difficult to draw conclusions from a comparison of studies covering different countries, species, and habitats, there is a need for studies on the effects of these linear artifacts on Australian wildlife. That's a quote from Andrews A. in The Australian Zoologist. He wrote a paper covering the effects of highways on biodiversity in Australia, and since it's not so readily transferable to other habitats, we found it might be important to see if a similar study in Michigan would yield similar results. So our question was, how do Michigan interstates affect biodiversity of tree species in an ecosystem as measured by the Simpson's Diversity Index compared to non-highway impacted ecosystems? Our null hypothesis was that there's no significant difference between these two zones, highway and non-highway, and the alternative hypothesis was that there is a significant difference between the two areas.
So we're gonna go over some criteria for our sites. First thing we're gonna go over is the criteria for our highway sites. There are a couple things that we really wanted our highway sites. We wanted a forested area with a clear tree line that wasn't backed up to an urban center like a suburban town or like a huge city or anything like that. We didn't want it backed up to anything that was overly busy. And we also want it clearly divided by a highway, which meant that trees on both sides, like the picture on the slide. We also want to be able to stop safely there for collection. That was a big thing, was safety. We didn't want to stop at the edge of like a 4 or 5 lane highway. That would just be absurd for us. And we wanted it more than 0.1 miles long, which means that so we had a larger area that if we didn't like one site or it was maybe too hard to get to, we could just walk a little further down the road and we could take another measurement. And now some selection criteria for rural sites. We wanted some of the same things: a forest area, clear tree line, wasn't backed up to urban center, suburbia, that type of stuff. We wanted away from major roads rather than a major road right next to it. We wanted, because of this, we thought that it would be a relatively preserved area of land, and we get a better sense for the original Michigan biodiversity that we'd be comparing. So now going through some of the actions at each site once we got there, and this is the actions that we took for both the rural and the highway sites. As soon as we got to a site, we identified the tree line and measured a 100-foot transect at that tree line. We then broke that 100-foot transect into 10-foot long transects, and we marked each 10-foot transect with separate flags. This made data collection much more easier, as we could we could sort the trees into the different transects. We've then photographed the bark of the trees. And the way we identified what trees needed to be photographed, it was basically 1 foot away from the transect line and in both directions on either side of the line.
Because we took so many pictures of trees through all of the sites, we had to label the data carefully. So how we label the data, we had a transect number, a specimen number, and a site ID. So for example, we'd have rural area 1, specimen number 1, and transect number 1. So this allowed us to ID each tree quickly and keep track of where they were. Then we could organize the trees later into their respective transects into folders.
Okay, so just a little bit about our identification process. Because of the season that we were working in, we weren't able to look at anything other than the bark, really. So we took pictures of the bark for identification, and then we also took leaves for further identification, just to make that process a little bit easier. We also used many different databases in order to identify trees through their bark. And we also took pictures of the buds on the branches, because you can also identify trees in that way as well.
The statistical test that we did was a t-test on the average biodiversity between the 2 different types of sites. And we did a t-test because we had 2 categorical groups, which was highway and rural, and we were trying to find the significance of the difference between the biodiversity of those sites. And biodiversity is a continuous value. And we found the averages for both of those types of sites, the highway and the rural sites, and plugging into a t-test, we got the p-value to be 0.359, and with the alpha value of 0.05, there is no significant difference between the biodiversities of these 2 groups. There was, however, way more variance in the highway sites than in the rural sites.
Okay, so in our discussion, based on our results, we can conclude that Michigan highways do not greatly affect the biodiversity of the area. This differs from prior research that we've looked at, considering that they indicate that in most areas of study, highway presence has significantly impacted biodiversity and disturbed the ecosystem involved. Given that our results deviate from what is generally expected, we might suggest that Michigan conservationists place efforts in another area. Possible errors include incorrect identification of trees and then transect measurement. Okay, so our takeaway message is, even though human structures have an undeniable negative impact on ecosystems, we were unable to find a significant difference in biodiversity between interstate impacted forested areas and non-interstate impacted forested areas in Michigan. Thank you.